bending upwards than bending downwards. And that's because in order to maintain these so-called GSAs, gravitropic set point angles, roots have to be able to bend upwards as well as downwards. These are the lateral roots, have to be able to bend upwards as well as downwards to maintain the GSA. So you can see a root here that's been reorientated. These roots have had to bend down, these have to bend up. So we've been really trying to understand what is the mechanism that's controlling that. And this is some work that was published last year. And in short, what we've shown is that as well as a gravitropic response in the lateral root, that importantly is angle dependent, in that as you increase the angle of the root away from the vertical, you get an increased magnitude of gravitropic response. That's counteracted by what we've called an antigravitropic offset, so a constitutive upward flux of auxin out of these columellar cells. And that's all controlled in terms of the localization of pins through a sort of antagonistic phosphorylation of those pins that seems to be where RCN1, the phosphatase RCN1, is incredibly important. So the underpinning data for that are over here. This is pin 3, and what we see in lateral roots is a bipolarity of localization. So we've got pins localizing both to the upwards and downwards membranes. So that's the context, really, this idea of antigravitropism counteracting gravitropism. So we wanted to know more about this. So we did a variety of screens. One of them identified this very interesting mutant, which we called lazy4D, because it has a mutation in what we have called this extended domain of domain 3. So when I refer to D3X, that's a little bit of additional domains on the end of domain 3. And that's only found in lazy2 and lazy4 in Mio's lab. Lazy4 would be lazy3, just to clear up that. And you can see here that this is the wild type, here's the lazy4, much more vertical root growth. Interestingly, lazy4 is also dominant. So this is in the heterozygous condition, very dominant. No effect on expression levels of lazy, gene expression levels in lazy. And this is a very highly conserved domain. So here you can see across many angiosperms and that we've got very clear conservation. And this arginine here is the one that is affected in the lazy4D mutant. And here's an AlphaFold2 model of lazy, not showing really very much because it's largely intrinsically disordered, but this is where our mutation is. And it's got some interesting phenotypes. I should say that we worked an awful lot with lazy2 just because it's slightly easier to work with. If we introduce the same mutation into the lazy2 background, then we can we get this same phenotype. And you can see here, these are these reorientation assays that we do an awful lot. This is what happens if you reorientate wild type roots. Some have to bend down, some have to bend up to get back to where they were. You can see that the lazy4D mutant has this more vertical phenotype, but it's perfectly capable of maintaining its GSA. Intriguingly, there is 0 primary root gravitropism phenotype in lazy4D, which I'll come back to later. Oh, and here's all the animations that were meant to help you understand all of that. Okay. So how is lazy 2 or 4D inducing this steeper GSA? Well, one of the things we notice is that in lateral roots, when we're looking, there's a shift, a downward shift in polarity in the lazy4D here. You can see that quite clearly. The other thing is that there is increased targeting to the amyloplasts. So this is the work that Mio and Haodong have just been describing. You can see these are the lines, by the way, the lines from Mio. Thank you very much for these lines. This is the targeting you see to the amyloplasts in the wild type lazy4. In lazy4D, we've got much more targeting to the amyloplasts. But what it doesn't do is affect pin polarity in this bipolarity of pin 3 and pin 7 that we see in the columella. So this is the reason for looking at that is that that's the basis of the model that we're working to. That's how you are able to maintain these non-vertical GSAs. So we don't see any effect of on pin polarity. That may make some sense in the context of these data, which show that in this case, lazy 2D doesn't require the RLDs for its effect. Again, thank you to Mio for providing those lines. So with this in mind, we've begun to think about what could be going on. We've got a columella process. That's where lazy is being expressed. That's where lazy 4D is expressed. No, it's not pin polarity. Is it activation? There are two possibilities here. It could be more of the down pathway, or it could be less of the up pathway in terms of auxin distribution. Because what's happening, what we need to remember is because lazy 4D is maintaining a GSA, it just means the equilibrium point at which the amount of auxin exiting the lower side and the upper side of the columella cells is equal. Now, just because I'm just naturally optimistic, tend to look at the upside, and I'm telling you a lot about how roots need to bend up, we're thinking it's more to do with what's going on the upper side of these gravity sensing cells. That's partly because of the polarity shift I just showed you. So there's this downward shift in lazy 4D. And also just to think about what is the evolutionary significance of this D3X domain? What would the world be like if we didn't have this D3X domain? Because it seems to be important for permitting this more lateral horizontal growth, or more horizontal growth. So if this wasn't here, we'd have root systems that were very vertical, not really able to forage for nutrients, which in an agricultural context is fine because we give our crops nutrients, but in nature, not so much. So we started to think about activation, and we were thinking about D6PKs. We have some very old data on D6PKs. Thank you to Klaus for the lines way back. And interestingly, Yiji's group have also recently shown that there is lazy-dependent recruitment of D6PKs during gravity response. And I was reminded of how the sort of really impressive phenotype that we see from the D6PK mutants. So here are two D6PK triples. There's the wild type. You can see they're really very much more vertical. Interestingly, just like lazy 4D, we don't see any primary root gravitropic phenotype in any of these mutant combinations. Yet we have this really significant lateral root phenotype. And so we looked at, we use a clinostat. So I haven't mentioned clinostats yet. It have been mentioned today. So a clinostat is something you can use to deprive the plant of a stable reference to gravity. And this is indeed how we came up with the entire model using clinostats. And this is what you would see. This is what you see in a wild type plant. Pre-clinal rotation, they're growing at their GSA. And then after clinal rotation, these roots bend upwards. And what's striking is that we don't see any of that happening in these D6PK mutants. So there's maybe a hint of something going on here in this double, but certainly over here in these triples, we've completely lost this upward bending. So for us, this is all still quite speculative, really. But this is an elaboration of the model I showed earlier. What we have is a system that's controlling the bipolarity of pins in the columella. And so that's really based on what we would refer to as the PID-WAG phosphorylation sites in the pins. The paper I described at the beginning, that has all the data showing that it's about those sites, and we have phosphovariant data from Yiji's lab that supports that idea. So there's something that sets up this bipolarity. We also have lazy 4D, which seems to be doing something that, as far as our interpretation is concerned, we think it's not able to do something, it's not able to service these upward pins to increase aux influx to the upper side. And currently, based on the data I've just shown you, the most likely explanation we have is that D6PKs have some role in that, based in the, I guess, on the circumstantial evidence that's accumulating from the people in this room. But it's really, I'd encourage us to think about this domain. I think it's a really fascinating domain. It's just a short domain. And I'm quite keen to, one of the reasons I desperately wanted to come was because I think there's lots of scope for collaboration to explore these ideas more. So I will finish by thanking again Adam and Suruchi, but there have also been important contributions from Marta, Martina, Faye, Ryan and Alex, and to other collaborators, Yeji, long-standing collaboration on GSA control, and also to Mio and Klaus in particular for sharing lines. Thank you.
Questions and comments? Okay, please.
Yes, Stephen, nice talk. Yeah, so I have two questions. The first is the point mutation. If you change it to different amino acids, will affect, or you need to specific amino acids?
No, you're absolutely right. I should have pointed that out. It can be anything so far. It seems to be not arginine is the important thing. And we also have a mutant in wheat that has the next arginine along that's changed to a tryptophan. So there seems to be some scope for dialing in different growth angles here. But yeah, it seems to be that domain. There are other sites. There are other sites that we've looked at in that domain that seem to have the same effect.
Okay. So the second question is the slides before this one.
Yeah.
Yeah. So you show the unphosphorylated pin proteins. It's worked and important for the positive gravitropic, right?
Yeah.
Yeah. This one. But yesterday we have discussions and reports from all these group. They show that really the phosphorylation is important for pin's activity. But can you comment how this unphosphorylated pin works?
Well, it's really, I mean, that's why I've had to add things in here. I mean, the sort of pattern of pin phosphorylation is probably quite complex. So I'm focusing here on something that's determining upper, lower polarization. On top of that, there are other phosphorylation events that other people are working on that we need to understand as well. So that is slightly misleading, and I will think about how to describe that. But this is basically this fundamental polarization question with respect to those PID-WAG sites.
Okay, so the general idea is some phosphorylation is essential, others not for the activity.
Yeah, in a more I have a more complex model in which there are multiple there's a site X and a site Y, if you like. This is the regulation on site X. So unphosphorylated is giving you this downward, phosphorylated is giving you this upward. And actually that's a general point I think I would make. But maybe I won't make now.
You can continue.
Okay. But some of these things I think are, if you're trying to think about the cell biology, the observed cell biology. So even things like how long does it take pins to relocalize after gravity stimulation in the primary route? It could be that really what we're looking at are things that are much more important in the context of lateral routes. So this longer term, so lateral routes, the other thing we need to remember is, of course, is that lateral routes are, the point here is that they're outputting a gravitropic response the whole time. So that's interesting in the context of how, Dol, your talk about phosphorylation and some of the questions, how is that constant gravitropic response being maintained over days, essentially? That's more vertical, but we have, so that fits, but we've had some problems actually identifying which ones are being expressed in the columella. That's one of the issues. So we don't talk about kinase very much because we aren't exactly sure which kinase it is.
But I mean, now that you have the D6 kinase phenotypes, do you need PIT-WAX in your model?
Well, it's the lines, the phosphorarian lines you gave us for the pin3 with the D6 sites changed. We didn't get any phenotypes from those. So as I'm seeing this as a model, we're seeing a polarization question, and overlaid on that is this activation question, which is where I think the D6PKs would come in. I can't rule out D6PKs being important for the localization.
But there is maybe a misunderstanding on your side on the relative contribution of the sites. I mean, the sites that we describe as being preferentially phosphorylated by one or the other type of kinase, but ultimately all of them contribute.
Yeah.
So you need the what you may call the PIT-WAX sites also for activation by D6.
Okay.
So all of them are targeted.
Yeah.
So there is no exclusivity.
I think the point is that I think there's two separable things. There's polarization and activity, which is uncontroversial in the context of this whole meeting.
Last question.
Thank you very much. I may have missed the point, but did I get it right that the D6PK only affects the bending angle of the primary root and not of the lateral roots?
No, we don't see any effect on the primary root, but we see this huge lateral root GSA phenotype. So they are much more vertical, and also they don't seem to be able to bend upwards in the same way.
Do you think this might have to do something with timing of the response? As D6PK come very early, or is it just a completely different system, so to speak?
I mean, there could be differences, but I don't think it's that. I think fundamentally D6PKs seem to be contributing somehow to this upward movement, this upward aux influx. That's one of the most obvious things for this. So I don't think it's a short-term, long-term.
Thank you very much.
Okay, thank you very much, Stefan.